Hi, everybody. Everyone got lunch? So I'm Phil Marsden. I'm a kidney doctor, but I spend a lot of time grinding up fruit flies and worms. So I take care of kidney patients, but at its heart, we're sort of interested in what goes wrong in patients with disease. So my talk today is a generic one. It is open. I want to encourage questions. I'm going to pick on members of the audience intermittently. I'm going to start with you, but not right now. Just making sure you got your game on. You too. You're second. All righty. So interrupt me. Questions. I'm probably going to talk about 30 to 40 minutes type thing. Does that work for everybody? Okay, gotcha. All righty. So let's start with a story. This is a seal. This is the Weddell Seal, and it's a fascinating beast. It's not endangered. There's about 800,000 of them alive, but they live in the Antarctic. Yes, and polar bears don't eat them. Why do polar bears not eat them? Polar bears don't eat penguins either. Why do they not eat penguins?
Because they live in different poles.
Yes. Yes. Yeah, polar bears will live in the north up pole, right? I mean, they're not on the southern. Penguins, though, sadly, the Weddell seals and the penguins can be food. So this beast has a really interesting property as a cardiovascular mammal that lives in the water. Firstly, it's absolutely huge. The males can be like almost 1,500 pounds. They're huge. They're not little seals. And they live off ice fields. And when they hunt for their food, they have the ability to dive for up to 1 hour underneath the water. So their cardiovascular system is much like ours. They go to incredible depths. Look at that, 1/2 kilometer. And they need blowholes to come back up. The only time they get into problems is when they dive and they can't come back up for that eventual air that they need. So how do they do it? What allows this organism to take a deep breath and go down to 1/2 kilometer for almost an hour? So here's a bit of studies on them. This is quite old work. It's 30 years old, and it shows a variety of measurements in these animals. For instance, the heart rate and the depth are shown on the left-hand portion of this slide. And what you see here is the animal dives very deep. See, look, this is on the right-hand axis. This is meters. So it's going down to like 1/4 kilometer, and then it comes back up. And this is a very prolonged time. And look what it does. Its heart rate slows down dramatically compared to us. And just as it's about to surface, not before, all of a sudden its heart rate goes way up. Look what happens to the concentration of oxygen in its blood. It starts off quite high as it takes those deep breaths, and it falls really, 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 really low. And as it starts to come back up again, it takes its first deep breath. What allows it to have some oxygen utilization during this period? And it's a fascinating organ. What happens is its spleen contracts and keeps all this oxygenated blood and squirts it out at it. It's a hugely interesting organism. I want to tell you today that what I think allows this seal to do that is its closed cardiovascular system. Humans have a closed cardiovascular system, but what is that? Is the title of my talk today. And my objective is to talk about the implications of an open versus a closed cardiovascular tree. And I want to share with you a little bit of introductory information on how endothelial cells of the closed cardiovascular system work and allow this Weddell seal to do this type of amazing physiology. Galen was a physician born in Roman times. He was of Greek extraction, and this is how he thought the cardiovascular system of humans and a seal, by inference, would work. He thought that the heart was the source of the energy which would pump blood, and that the blood would be sent out to vital organs such as the brains to allow us to think, or the kidneys to allow us to make waste products. But that once the blood got to the brain and the kidneys, it would get consumed and used up. What Galen thought 2,000 years ago is that the blood was made by the liver, and when it got out to these organs, it was consumed. It was open. It did not recirculate. So every time the heart beat, the blood went out, but it did not come back and had to be made afresh. And the amazing thing is, this stood for over 1,000 years, this model. And it was only in the early 1600s that William Harvey said, no, 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 no. This is how the system works. The heart is still the pump, but now the blood that leaves the heart heads up to the organs such as the brain and the kidney, gets used there for nutrients, puts waste products in, and then it recirculates back to the heart. So how did William Harvey, who discovered this in 1628 in terms of the published work, figure all of this out? Well, he did both thought experiments and top of the wet bench experiments. His thought experiment was, well, the human heart beats anywhere, let's say 100 beats per minute. The volume of the blood in each of the left ventricle as it squeezes is, let's say about 50 or 100 mLs. And he reasoned then that there had to be almost 5,000 ml or over a gallon pumped by the heart every minute. And he said, I can't see the liver producing a gallon of blood every minute. So that was a thought experiment, and intuitively of interest. What he did at the wet bench, though, is he put tourniquets, sequential tourniquets, over patients' arms. So he had subjects, and he would put a very tight band. And he would put a band that was really, really tight, and it obstructed all blood flow in the arms, so there was no blood getting in there, let's say, above the arterial pressure. So the hand would get limp and numb. It turned bluish, whitish a little bit. Patients would report weakness. They'd have tingling of their fingers after 10 or 15 minutes. And he couldn't feel any pulses. The other thing he did was he put up tourniquets below the arterial systolic pressure, but above the venous pressures. And what that did is it let the arm get swollen and bluish. He could still feel the pulse in the radial artery a little bit, but he reasoned from that by releasing these tourniquets and looking at the veins on the top of the arm and raising them. He figured out that the artery blood, the bright red blood, was going from the center to the periphery, but that the vein blood, the bluish blood, the deoxygenated blood, going back on the top of the arm that he could observe, that blood flow was going from the hand towards the heart. And that allowed him deductively, based on this wet bench type experience, to say the blood flow was in a closed cardiovascular system. And that's what we now appreciate. So if I'm comparing a closed and an open cardiovascular system, which we'll do, let me just start with the most important thing, that no flow is the worst thing. To get an organism bigger than a millimeter inside, you really have to recirculate things because you can only rely so much on diffusion. There's a few parts of the human body where diffusion occurs and there's no blood flow. Does anybody want to hazard a guess? I'll take this man here in the white.
Can you repeat the question?
Well, actually, you could, you can see this issue in our eyes. Our corneas on the surface of the eye and the lens of our eye have no blood vessels in them. Everything's through diffusion. And an animal that doesn't have that capacity, like the sea cow, the manatee, has blood vessels of its cornea and its lens. It can't see properly. Humans who develop blood vessels within on the top of the cornea can't see properly. But there's a ton of diffusion in our lenses. Why? Because we need the light to go through and obstruct it. But no flow is the worst thing in a higher organism. And a good example of that is when we use kidney organs to donate from donors, either living or deceased, and give them to people whose kidneys have failed and we want to replace them, we couldn't figure out how to transport the organ from Kitchener to a patient at St. Michael's Hospital and do the kidney transplant. When I was doing my training, we would compare this fluid with that fluid, bringing them on ice over, bringing them with fluid over. But it took a bit of time for us to figure out that the most important thing is to keep the organ perfused. And it didn't matter what we perfused it with. You just had to have flow over the circular of the blood vessel. So now when we transport organs, the kidney is put in a little perfusion system where there's flow of liquid going in the artery, coming out of the vein. And we now know those kidneys do much better on the short and long term. So what is an open cardiovascular system, Phil, and what is a closed one? An open cardiovascular system is where the blood, let's call it blood for now, flows through open spaces. We could say, well, Phil, why did you call it blood? Well, you probably shouldn't, because the cells that form part of our organs in our body have fluid around them. Let's call that interstitial fluid. That interstitial fluid in humans is different from blood. But in organisms with an open cardiovascular system, it's the same fluid. So technically, it's called hemo for blood and lymph for fluid, hemolymph. And it flows into open spaces. So it comes out of a pump, akin to the heart, and then it goes out into an open space. And that open space is called the hemocoel. The blood and the interstitial fluid mix. But here's some of the downsides of this system. The blood flow is very slow. So it eventually does come back to the pump through these holes, but it's a very slow system. The internal organs are all bathed directly by blood. So if you're a cell, let's say of the liver equivalent or of the kidney equivalent of a lower species, you are bathed by this hemocoel stuff. The downside of that is that the delivery of nutrients and removal of waste products is very, very slow. And it's really hard to regulate all that stuff. Well, which organisms have an open cardiovascular system? And two of the largest phyla are arthropods and mollusca. This is an example of an arthropod, a lobster. A lobster has an open cardiovascular system. But boy, are they delish, right? This is a horseshoe crab. It's technically not a crab. It's more closely related to a spider than a crab, but you know, the word crab stuck a long time ago. This is a fascinating beast. They can get very large. It also has an open cardiovascular system. It has a very weird hemocoel. It has a very weird hemolymph because its blood, when you look at it, when you're just staring right at it, it's sort of like a opaque white, maybe yellowish, but as soon as oxygen in the air mixes with their blood, it turns green. Their ability to carry oxygen in their hemolymph is through, not hemoglobin, but hemocyanin. And they're a very interesting beast. I'll tell you just a couple of things about them. Firstly, they swim upside down, which is like, that's dumb. So they swim upside down at an angle, and they're pretty fast. They are commercially harvested. And they're commercially harvested because this hemolymph they have has white blood cells like our body does to fight off infection, but they're called amoebocytes, and they're very, very sensitive to bacteria. So in clinical medicine and in bench-based research, we take those amoebocytes of the horseshoe crab and we use it to measure waste products and degradation products of bacteria to see if anything's contaminated with bacterial products, endotoxin. It's the basis of our endotoxin assay in research to look for infections. What a crazy little beast. This is another example of an arthropod, an insect. An insect also has an open cardiovascular system. This is a photomicrograph of the open cardiovascular system of that mosquito. Though it sucks our blood, its own blood is completely different. It's hemolymph. This is the staining of its pump system that pumps its hemolymph around its open cardiovascular system. And these are the surrounding cells. We call those pericytes in adult human biology. This is what the heart tube of a mosquito looks like. So arthropods and molluscs have an open cardiovascular. Molluscs, a good example of those would be bivalves like scallops or clams or oysters. Not every mollusk has an open system. For instance, squid and octopus are in the family, the phylum Mollusca, but they have a closed cardiovascular system. So these older definitions and groupings that were started off many, hundreds of years ago, as we learn more about physiology, they're not totally intact, but they've stood the test of time. The closed cardiovascular system differs in that blood flows through closed blood vessels. The blood flow is much higher, and the internal organs are not in direct contact with the blood. The advantage of that, and one of the advantages for the Weddell Seal, is that the delivery of nutrients and removal of waste products is really rapid. But in contrast to the open cardiovascular system, which is hard to control, this statement is not correct. It's easy to regulate blood flow in the closed cardiovascular system. And I want to chat a little bit about that. So this isn't a sea mammal. This is an elephant. It has a closed cardiovascular system. It has a very elegant one. And if you look at huge species like elephants or giraffes, their closed cardiovascular system is really interesting. Their pump has a lot to deal with. For instance, their heart can hold 2 gallons. They have to pump their blood all the way up to their head. They're vertebrates. They have the same neck muscle structure and bone structure as ours. We have 7 cervical vertebrae, so do theirs, but each of theirs is 11 inches long. Their closed cardiovascular systems work the same as a very small beast, like a little mouse or a shrew. This ugly-looking thing is one of the oldest, oh, good for him, he knew it. It was a hagfish. This is one of, species either extinct or extant. This fish, known as the hagfish, is one of the, interestingly enough, one of the most primitive species that's still on the good earth that has a closed cardiovascular system. And it evolved about 540 million years ago. It's not called a jawed vertebrate, even though it has a jaw, so I don't get all this stuff. It turns out a jawed vertebrate has 2 jaws, but this one has 1, so it doesn't count for some reason. This is a really interesting fish. This fish, when it's startled or agitated, from these little outpouchings on the side of it secretes this mold, this slime, that mixes with the water, and it can produce 20 or 40 liters of this slime mold immediately that squirts out of it to protect itself against its prey. When it does that, it has this very weird habit of tying itself into a knot and pulling the knot along its body to get away from the mold, but the predators that are after it can't do it and get suffocated, especially if it gets around their gills, if they're gill-breathing fish, for instance. There's not many commercial purposes for hagfish. There's only one culture that eats them, ugly-looking fish, the hagfish. But it's really interesting because that's one of the oldest species in evolution, an extant one versus an extinct one that has a closed cardiovascular system. So what's going on at the microscope level in Harvey's model of the closed cardiovascular system? And the big thing I want to just highlight very early is the heart does the same thing in both systems. It's the pump. What's unique to the closed system is that there's an interconnection between vessels that leave the heart and ones that go back to the heart, these microvascular capillaries. And in humans, we have over 100,000 kilometers of capillaries that allow the blood to recirculate, where often at the single cell level, a red blood cell of humans is going through a very small architecture. The really fascinating part of this whole system is that as the closed cardiovascular system developed in evolution, the cells that line the closed cardiovascular system were born. So the hagfish has a lining of its closed cardiovascular system called the endothelial cell, and the heart with the endothelium are the 2 key cells of the closed cardiovascular system. And that's actually what my lab and my students, many of which are here today, work on. One of the things that co-evolved just before the closed cardiovascular system was the ability to carry oxygen from the environment to the cells that needed it. And this is one of the key molecules that allowed that to happen. This is the heme structure around which oxygen interacts with an iron atom in the middle of the structure. And in hemoglobin, there's 4 proteins, 2 molecules each of alpha and beta globin that wrap around each of these things. So there's 4 hemes in 1 molecule of hemoglobin and 2 alphas and 2 beta globins. And if you're a kidney doctor, the proper function of hemoglobin is really important because the kidney cells produce things that stimulate the production of red blood cells that contain heme to enhance the delivery of oxygen into the periphery. So that Weddell Seal, that within its spleen was holding on to oxygen-rich blood, and when it was really, really deep and it needed more oxygen, it would contract its spleen and send that oxygenated blood back out to circulation to give it a little life supply of oxygen. The horseshoe crab, I described a little earlier, does not have iron. It has copper as its ability to interact with oxygen carried around its body. So these are these little red cells that circulate within our body, some 25 billion of them at any given time. And they're the beasts that carry the oxygen from the lungs to all of the tissues of the body, and from the body carry the carbon dioxide back to the lungs to be expired. So one of the very cool properties of the closed cardiovascular system was the ability to differentiate. And here's what happens when the blood's coming back from the systemic circulation that's lost its oxygen because it was used, that's gained CO2 because that's a waste product of the breakdown of glucose. And here's the exchange network, the capillary of the microvasculature of the lung that allows that reoxygenation to occur. The closed cardiovascular system took advantage of all of the nuances of the physiology and started to adapt. And this complex slide is a structure of the right and the left side of the circulation of the heart. And it's interesting to note that we don't allow it to mix in adults. It's sort of crazy, but that, I'm going to come to in a second, but that when we're in mom's womb and we're developing an organism, we do not have lungs. So we don't breathe in air during, during the stage of being in utero to allow oxygen-rich blood to come to the left side of the heart and get sent up to the kidneys or the brain. So that creates a dilemma because the blood's coming back from the extra-embryonic tissues of the baby, goes back into the right side of the heart, but there's no reason to send it to the lung. So during development, we shunt the blood across to the left side of the heart. One of the ideal ways is there's a hole from the right atrium to the left atrium. That's called the foramen ovale. That's supposed to close as we get to, as we take our first few breaths. And there's an additional connection between the pulmonary artery that comes out of the right ventricle and the tip of the aorta, the ductus arteriosus, that allows blood to come from any that got squirted out of the right side of the heart to go over to the left side of the heart. Because that's key, because that's where the oxygen's coming from in utero, because the oxygen has come from mom and the placenta. Anything that messes up this circulation in an adult is a really bad thing. So does anybody want to hazard a guess about what would happen if blood from the right side of the circulation was shunted over to the left? Do you want to try that? Said I'd pick on you. What color would the blood be? So instead of the blue blood getting reoxygenated in the lungs of an adult, now that blue blood gets sent out into the artery, so it's going to dilute out the oxygen carrying capacity, isn't it? So your ability to transport oxygen gets markedly mixed up when you have a right to a left shunt in, for instance, a baby with congenital heart disease, which is the most important developmental abnormality in newborns in Ontario. Each organ has a very complex closed cardiovascular system that puts those capillaries together. I'm a kidney doctor, and I'm amazed at all of the different microvascular nuances of the kidney. One of the kidney's key jobs is to take the blood that comes to it and squeeze it. And it makes a filtrate, takes about 10% of the fluid out of the blood, and sends it out into the bladder. Well, that would be a problem if we couldn't reabsorb some of that, because we filter about 180 to 200 liters of fluid per day. So everybody would go in the bathroom all through Phil's talk. But we take out 99% of that fluid, and on a good day put out 1 liter 2 liters of urine. So all of this microvasculature of the kidney is all set up to filter the blood and to reabsorb all the salt and water so you don't put out 200 liters of urine a day. Geez, that's a lot, right? That's like a gallon an hour. The kidney does a lot of amazing things, but it's this microvasculature of the glomerulus that's lined with endothelial cells that allows that filtrate to occur. So it's letting a lot of fluid out. It's completely different than the endothelial cells that line the insides of our brain, which are very tight and don't let much out. In fact, doctors trying to give medications to patients with diseases of the brain, let's say an infection around their brain, the meninges, some antibiotics don't get across that blood-brain barrier. The really interesting thing about the kidney is it filters absolutely everything. But it's really good in just doing the small molecules. It doesn't like to put out things like proteins. And we see patients in kidney disease where the ability to not put out things like proteins allows the proteins to get out in the urine, and that's a disease called nephrotic syndrome. There's big vessels and there's little vessels in the kidney. This is the main artery that goes to each of our 2 kidneys. And just like in the heart, and just like in the aorta, you can get the chronic aging disease of advanced countries, and that's atherosclerosis of the closed cardiovascular system. And sometimes the blood supply is cut off so much that you develop dead tissue downstream. You develop dead tissue downstream if there are not blood vessels that are hooked up in a network that allow blood to come back in if one of the blood vessels is blocked off. In the brain, we're really good at hooking up the closed cardiovascular system, so if a little bit gets cut off, there's other blood vessels to go there. There's a circle underneath our brains, the circle of Willis, which is really good at supplying blood vessels if one of those gets cut off. But in the kidney, they're end arteries, and they don't anastomose, and they'll cause infarctions if you cut off one of them. Well, Phil sort of told you not the whole truth. There are parts of the human cardiovascular system where there is an open circulation, and it's important to know this. The placenta of the baby, the small blood vessels that are used in pregnancy, is an open circulation in higher species, including us. It's just to remind us that when we are a freshly fertilized egg and we start to divide, one of the first things that happens when we become a hollow ball is one little part of that hollow ball turns into a group of cells known as the inner cell mass that becomes the embryo, or us. The rest of that develops into extra embryonic tissues that supply a lot of nutrients and hook up with mom's circulation in the first little bit of life. And that is known as the placenta and the amniotic circulation, the yolk sac. So this is a sort of a generic picture of what the placenta of the baby looks like, hooked up in the uterus of mom's womb. These is the major vessels connecting extra embryonic baby tissues, the placenta, to the baby through the belly button. This is the umbilical cord. This is what gets tied off when the little bundle of joy is turned off and the placenta goes into the garbage. Sometimes we get endothelial cells from this cord because the umbilical veins and the umbilical arteries are lined by endothelial cells, and my research lab goes up to the labor and delivery suites, and we're able to isolate the endothelial cells off this and use them as a model of human endothelial cells. This is extra-embryonic baby tissue, this stuff here. This is mom tissue stuff. And there's things that go on there at those very early stages that we never see any time later in our life, because some human genes only function at this period of life. So how does all this system work? These are mom's arteries coming from her big uterine artery, and these spinal arteries here somehow allow oxygen to be sent to baby, and somehow the carbon dioxide generated by combustion within the baby gets sent back to the mom's thing. So how does that happen? So it turns out the blood comes from the spiral artery of mom. It's a normal closed cardiovascular system. That then it does something really interesting. It dumps it into a big lake. And in this picture, This is where mom's blood gets dumped by those spiral arteries. This is baby tissue. And there's these little villi, little feelers, that within it have baby blood cells. And surrounding that is a very weird cell type known as the syncytial or trophoblast of the baby, which is actually one big, giant, fused, multinucleated cell system. And that's the interface about which mom's oxygen gets transferred over to baby tissues. But in many ways, this is an open cardiovascular system of development. So it's sort of interesting that, I mean, we're a higher species and we have an open cardiovascular system within the placenta of our species. And it's a function of higher mammals, as you're probably aware. These cells that line the top of the little baby's villi are very good at exchanging nutrients. And they also produce some key hormones. They're the cells that produce human chorionic gonadotrophin, which is the basis of a pregnancy test. So these cells produce it, it gets into mom's circulation, and it can be measured there. But it's actually baby-derived. This is a schematic for those who are trying to follow that, because it isn't easy. So here's mom's spiral artery blood, lots of oxygen. Those are her endothelial cells, and then all of a sudden, these are the floating villi of baby circulation, which is lined by syncytial trophoblast. This is maternal blood dumped into these big giant lacunes, and that's where the interchange occurs. And you could say, well, if it's just a little floating villi, like, why doesn't it just come out when mom's walking around? Turns out some of these villi are anchored into the uterus wall, and their job is to hold on. And they got a tough job, because as soon as the little bundle of baby is born, the whole of that time is send lots of blood to baby, lots of blood to baby, so all those nutrients can get there and baby's growing really quickly. But as soon as the baby's born, one of the huge problems is now all of a sudden you have this tissue that's been oxygenated by mom, but it's not needed anymore. And now what mom has to do is sever that connection with the extra embryonic baby tissues and deliver the placenta so that mom and baby can be both independent. Can anybody see a challenge of that for mom? What could happen when that's happening? She could bleed. Because if that's not done properly, then all of that blood is going to massively leak out. And in developing parts of the world, peripartum hemorrhage is still one of the most important causes of maternal morbidity and mortality. Bleeding at the time of pregnancy. So it's an obstetrical emergency, postpartum hemorrhage. But, it's like the whole pregnancy is like get blood, get blood. And then as soon as the baby's born, it's like cut it off, clot the blood. But you don't want clotting when the baby's there. You want lots of clotting when the baby's just been delivered. It's a very important challenge, and those interested in diseases of females during pregnancy, it underlies a lot of the pathophysiology of one of the most important diseases of pregnancy that's characterized by high blood pressure, protein in the urine. Anybody know what that disease is? It is the most important cause of maternal morbidity and mortality in Canada. It's called preeclampsia, high blood pressure pregnancy. And I'm shocked that there's very little lay knowledge about this. I don't understand why the Heart and Stroke doesn't push this and teach us all about this important high blood pressure disease of pregnancy. So let's just get back to this as we're wrapping up. This is the closed cardiovascular system versus the open cardiovascular system. And it's really important for us to remember that it took us an awful long time in treatment of patients and understanding physiology to make that break between what was suggested in Roman times to what we now appreciate as the physiology. Who knows what in 100 years we will talk about that we didn't know right now. The system has a few very interesting features that I want to cover. I just want to remind you that here's the artery, here are the capillaries of this closed system, here are the venules and the veins as goes back to the heart. Here's some of the properties of this. The total surface area, now not the length, I did that 100,000 kilometers of capillary length, but if you calculate the surface area as you march down this tree, you see how massively it goes up in the capillaries? It's a huge surface area. Here's the flow of a red cell going through it, the velocity. very high in the aorta and goes crashing down the capillaries and speeds back up in the veins as it gets back to the heart. So when I'm on the wards with the doctors, I ask them, so what are the forces in that system? And one of the really interesting properties of clinical medicine right now, and what I was sort of alluding to, joking about the 100 years from now, what will we look back on, is the most important thing you hear about when you're in the intensive care unit is what's the pressure, the blood pressure. What's the blood pressure in the arteries? You know, because the heart contracts, it's higher during contraction systole and relaxes a little bit in diastole. So it might go 120 over 80, 120 over 80. If the heart rate's 60, that means every second it's contracting and relaxing. But if I ask the trainees on the wards, where's the hydrostatic pressure, the blood pressure, highest? Let's say you average it out over the cardiac cycle. Is it higher in the capillaries versus the artery? And they say, the artery, because you put a needle in the artery, it goes squirting all over. Then I say to them, is it higher in the artery or the vein? They say, it's higher in the artery. And then I ask them, so is it higher in the capillary or the vein? Then they think about it, and half of them are like, I thought about that. But you know, they're putting catheters into arteries and atherosclerosis. They're measuring the venous pressure all the time. And they know the capillary pressures are really, really low, so they end up concluding, the pressure is higher in the veins than the capillaries. And then I ask them a question which has always interested me, in that, wait a minute. If the heart's the source of the energy, how can the pressure go higher in the vein than the capillary? And that really gets to people, because when you start thinking about hemodynamic forces, it's hard to understand that. And you know, people say, I'm walking up and down, my muscles are pumping the blood. That's not the explanation. It turns out that the biomechanical forces that are exposed in the closed cardiovascular system are unique. But the open and the closed system, the compressive hydrostatic pressures that are generated with every cardiac cycle is the same in Galen's model or Harvey's model. What's very different is that as the blood circulates, the frictional shear stresses that are exerted by the blood on the endothelial cells are very unique to the closed system, and I want to just illustrate it. So this is a simple thing you can do in a high school science project, maybe even a public school project. You can take a length of tubing, put a big long constriction in it, and then have it open up again. And as a kidney doctor, we see this in the vessels we use to do hemodialysis on patients. It occurs in the coronary artery. Here's a big coronary artery coming around, and here's atherosclerosis, and then it opens up again. And then you say to yourself, so how do you measure that constriction? And a very common thing is to use Doppler ultrasound to measure the velocity of the blood within the lumen. So then I say to the trainees, I say, so what's the flow of blood here versus here versus here? And then they start thinking, velocity, flow, is there a difference? I don't know. But if I was to ask them on Niagara Falls, what's the flow versus the velocity? They might see a log going down the river, and they say, what's going that fast? And then I say, what's the flow? They realize that it's volume divided by time. But velocity is distance per time. So it turns out the flow is going to be identical here, here, or here. Otherwise, the system would back up. It wouldn't be closed. It wouldn't recirculate. But let's say that the diameter here is 2 units, and here's 1, and that's 2 units. If you do the math, it turns out the velocity of the blood goes from 1 to 6 to 1. There's a huge increase in velocity here. And that's what Doppler ultrasound measures. It does not measure flow. Flow, you'd have to know the diameter of this and the diameter of that to determine it. So it's very common to make that mistake in clinical medicine. But here's the really cool thing. If you measure the hydrostatic pressure in these 3 regions, look at what happens. It's high here, it's low here, and then it goes back up again. It goes back up again because the energy that it takes to go from 1 to 6 to 1, that's kinetic energy. And the total energy in the system's constant. The energy will fall because there's a resistance here. But it's really important to remember that velocity and the friction of that blood going over the surface here is a major component of the forces. And if we thought about the Don Valley, it's intuitive. It's like, where's the energy of the cars on the Don Valley? You'd say, it's the speed of the car times its mass. But we don't ever think of like the air around it and its pressure. I mean, that's what the hydrostatic pressure of our blood vessels is. And it's a really interesting part in clinical medicine. We do not talk about the inertial energy, and let's say we've got Mrs. Jones and she's sick in the intensive care unit. We don't have rounds saying, I think Mrs. Jones' inertial energy in her capillary system's too low today. We say, no, it's her blood pressure. So how does the body regulate this type of stuff when there's no disease? Well, it has a very cool pathway. Turns out the endothelial cells can auto-regulate blood flow. If they're not getting enough, they're not happy and they start making more. And the Nobel Prize was awarded in 1998 for the discovery that the cells that line the cardiovascular system can release a substance that causes blood vessels to relax. And that molecule is itself a simple gas, nitric oxide, one atom of nitrogen and oxygen, and it causes blood vessels to relax. It can happen within seconds when you increase flow, and it can be changed chronically over days, months, and years. People with atherosclerosis don't make enough of this, and we know that because nitroglycerin is what we use in the intensive care and the coronary care unit to regulate blood flow in patients who don't make enough of this. It also is very cool because it stops things from sticking. It stops blood vessel wall cells from overgrowing. And it's a really important component of how we respond to our environment. So I'm going to ask a question, and the question is, and it's on the theme of the Weddell Seal. Now instead of we're going down half a meter, we're going to go up above 4,000, 5,000 meters. So one of the questions I would ask is, what happens when humans go that high up? Let's say what happens on the long term. Well, the generation's completely shut down because you cannot reproduce above 4,000 meters unless you've done it for multiple generations. And there are people on the earth who have adapted to living in those very high environments. For instance, in the Andes and the Himalayas, there are folks who have lived there for many thousands of years, and they've adapted to it. And if you say to yourself, how have they adapted to it? It's way cool. So I told you the kidney produces a hormone that stimulates red blood cell production. That drug is known as, that compound is known as erythropoietin. And it's the same thing that's used by biathletes and cyclists and cheaters in drug doping. So I would ask y'all, what do you think, and let's say you lived in the Himalayas, what do you think the hemoglobin concentration is of someone who lives as a Sherpa there? You want to take that one? Yeah. Yeah, because I mean, that's what I was taught about erythropoietin and how it works, right? I like doing endurance sports, and I would love to jag myself up with some erythropoietin so I can beat all those young guys. But it turns out, one of the really interesting is the hemoglobin concentrations of the Sherpas in the Himalayas are actually lower than you and me. But if you go to the Andes, it's completely different. They do have a high hematocrit. Their blood's rich and thick. So how do you think they do it? So wait a minute, you're really high up, so the oxygen concentration is lower because the total particle pressure of water. So your environment's got low oxygen free concentration. Your hemoglobin concentration is lower, so you can't carry as much blood per unit volume. How do you think they do it?
They produce more nitric oxide.
Well, it turns out that's what they do too. But physiologically, how do you think they do it? Anybody? Okay, I'll tell you. So if you measure the flow in someone's arm, their blood flow at rest is 6 times yours or mine. Their blood that closed system is just whipping around as fast as it can. Not a little bit, but 6 times as much. And it's nitric oxide that allows that to happen in major part. I think that's a way cool phenomenon. This is the surface of a mouse brain. There's a vein in the background, an artery in the foreground, and this is an animal where the gene that makes nitric oxide is lit up. And you see that the arteries, the bright green apple fluorescence, is the gene that makes that nitric oxide to allow those Sherpas to increase the blood flow to their arms. It's especially potently expressed at arteries, and it makes the blood vessels relax and allow more blood flow to go up there. It's a fascinating beast. Veins have some, but nowhere near as much. It's the arteries that regulate this. I want to make sure I close with the following points, that historically clinical medicine is focused on the descending hydrostatic pressure, but focusing solely on the blood pressure within our cardiovascular system emits the most important difference between the closed and the open system, and that's that the blood recirculates and exerts frictional forces on our circulation. And the endothelial cells are all about that. This, I'm a clinician, and we look to the New England Journal of Medicine as our premier journal for big studies. It was really hard in clinical medicine to show what level of blood pressure control for people in high blood pressure you need to bring them down to prevent the long-term complications of high blood pressure, such as atherosclerosis and stroke. Just so you know about high blood pressure, for people over 50, how common is it? What's the prevalence? Anybody want to take a guess? This man right here? Well, take a guess. You think it's 1 in 100, 1 in 10? What do you think? High blood pressure. Yeah, this man right here in the, is it a Berkeley shirt? Yeah. Okay. Berkeley bound. Okay. What do you think? It's 55%. It's extremely common. So this is an important question. How do you treat people? And nowadays, this trial was the first one that showed if you bring them to a systolic target of 120, they do better than if you bring them to systolic of 140. Before that, there was tons of argument. And right now, the recommendations in the United States are still around 140, 150, if you look in the textbooks from last year. But one of the reasons it was hard to show this, and it took so many years and so many hundreds of thousands of patients, is focusing solely on the blood pressure misses the nuances of the closed cardiovascular system. And here's one of the weird parts of the closed cardiovascular system that isn't all good. When the blood recirculates, it turns out how the blood flows, its geometry becomes an issue. So when you get to bifurcations, branch points, and curvatures of our human system, that's where the disease atherosclerosis shows up. And if you send blood down a tube and then cause a bifurcation, you get very weird hemodynamics that aren't purely friction now, but disordered flow, where the flow is going to and fro, and it's chaotic. And you can model that in little parallel plates apparatuses, some members of my lab do. And that's one of the explanations for why the human blood vessel disease atherosclerosis occurs selectively at regions of bifurcations, branch points, and curvatures. It doesn't occur in flat regions. This is a mouse aorta. This is where the aortic arch valves would be. There's the arch coming out. They have 3 blood vessels on the top of their aorta, just like ours. This would then go down the tummy. This is oxygen-rich blood. Around here is where that ductus arteriosus connects the circulations in utero. These are the vessels that go to the neck, and this poor mouse got bad genes from its parents. It can't handle cholesterol properly, and it's on a Western diet with lots of cholesterol. And after a few weeks of life, you can, if you stain the blood vessels with a dye that's red and sticks to fat, you see tons of fat. This is what these blood vessels look like if you take them out completely. Here's the aortic arch. It's going down the abdominal, thoracic aorta, the abdominal aorta, it bifurcates into the common iliacs of our legs. And this red is robustly full of fat. That's where the chronic disease atherosclerosis occurs. This is the downside of the closed cardiovascular system, because the high speeds necessary to deliver the nutrients to that Weddell Seal and efficiently hold on to oxygenated blood and exchange it, the downside of those physical forces exert lots of hemodynamic insults. You can see this. This is the aortic arch, cut on the one side and layered down like a flat book in front of us. Here's where the valves are. The blood's going to the top. This is the top of the arch. This is the greater curvature, the inner curvature. Early at 4 weeks, later at 12 weeks. Look at all that fat. That's full of macrophages. The human equivalent of the amoebocyte, that's full of fat within patients with cardiovascular disease. My goal today was to talk about a closed cardiovascular system and answer the question, but what is that? I wanted to understand with you the implications of an open versus a closed cardiovascular system, and we use some examples from nature to talk about that. And I wanted to talk about how the endothelial cells that line our closed cardiovascular system work. Thanks for your attention. Let's have some questions.